and we go way back to graduate school days. So just a little bit of Charles's history, but as you can see, he's at Mount Sinai School of Medicine in New York City, where he's a professor of neuroscience, endocrinology, and geriatrics. And that, I think, statement right there tells you he's a broad-based scientist considering a lot of problems, as is also clear from his title of the talk and what he's going to present to us. But one thing also interesting, particularly interesting about Charles in this presentation is that this is kind of full circle for Charles. He may be talking about this, so I hope I don't steal your thunder too much. But I knew this before, but I didn't know all the details. I'm not going to go into all of them. But Charles really had his scientific birth, if you will, right here in this building in the Department of Pharmacology, where as a high school student, he came and did rotations, if you will. I think in Dr. Stavanoa's lab, who was pharmacologist at the time, and maybe a few other people. But it started here, and it's not ending here, but it's nice to have him here to tell us kind of midway through his career where he's at. Anyway, after San Antonio, he went to Boston, where he got his bachelor's degree at MIT. And while he was there, I asked him today, how did you get into aging? And back in the day, there weren't very many people who were getting into aging, and the access points were fewer and far between. But it was a book, and I know a couple other people who got their start by reading a book called Time, Cells, and Aging by Bernard Strahler, which was one of the early reasonably good treatises on theories and data on the biology of aging. And he got really fascinated by that question because he wanted to do something he felt that was going to be interesting for the rest of his life. And that led him to USC, where Bernard Strahler was, but also a guy named Caleb Finch was there, and just starting his career. He'd been at Rockefeller and came out west. And so he ended up working in Tuck Finch's lab. I happened to already be there, and that's where we met. And so I got my Ph.D. I was a little slow. It took me a long time, not typical for me. About 1982, I got out of there. And he got his in 84, much quicker turnaround time. Then he went to Rockefeller, and he followed his neuroscience leanings and worked in the lab, I believe it was of Donald Pfaff, a very well-known behavioral neuroendocrinologist. And he's never left New York, though he moved on to Mount Sinai fairly shortly thereafter and has been very successful. So Charles' work, as you can see by this title, spans a lot of interrelated processes, from diabetes to obesity to aging and dietary restriction. And he's going to talk about his efforts and successes in trying to link these to common regulatory mechanisms. The only other thing I'd like to say is he's a great teacher, as I think you'll see. He's got tremendous energy and excitement about what he does, and he's always finding new ways to attack a challenging problem. So Charles, thanks for coming.
Well, Jim, thanks, first of all, so much for inviting me. I consider this place to be the mecca of aging research. And it's a strange fact of my life that I grew up here, but then I wound up not working here, because this is where I really should be. So really always a great pleasure to be here, and especially to be able to present my work. And so again, thank you, Jim, and thank you for that very nice introduction. We do go way back. And I hope I'm only halfway through my career, but I guess it may be not that soon before I'll have to retire. So anyway, so you see my topic. I would just like to point out that I hope it's not as broad as it sounds. There's a certain possibility in my research I have been criticized for being a little unfocused. To me, these are really all the same question, and hopefully I'll be able to convince you that that they have a lot of things in common. But first I want to very briefly address a question which I think is always sort of in the back of our minds. And maybe I don't really need to dwell on this on this point in this audience. But there really still is a lot of skepticism about whether there really is such a thing as aging. This is a very famous article written in 1997, but still a lot of people would argue that there is no such thing as aging, that it's just a collection of various processes that take a while to unwind. And in particular, in the case of cancer, you need a lot of mutations to occur in order to get cancer, but it just takes time. And what does that have in common with Alzheimer's disease or normal age-related impairments? So, but they make a point here that I would like to emphasize. They say, and yet, there still remains a strong and mechanistically unexplained relationship between the lifespan and the rates of these processes. And they're talking about these processes being cancer, but same thing could be applied to any disease of aging or any age-related impairment. There is this strong link, and that's actually what essentially propelled me into aging research, because there is a link between lifespan and age-related impairments. That the impairments and the diseases come on at the same point in the relative lifespan, regardless of 2 or 3 orders of magnitude difference in the lifespan. So in other words, with cancer, you get it about a little bit after halfway through the lifespan, whether you're a mouse or a person or C. elegans or flies. So there is some kind of a strong link, and that link is what I think we're trying to address. Furthermore, and more pertinent for this audience, it's not just that genetically there's a link. It's also the case that if you manipulate lifespan, you do wind up manipulating the age-related impairments and diseases, especially with dietary restriction, which is going to be a big part of what I'll be talking about today, but also with temperature, by cold temperature. So there's a very strong link between age-related diseases, age-related impairments, and lifespan. I think that link is actually aging, and I will try to address that link to at least some extent. Okay, so first of all, we all know this, but I think we don't perhaps completely appreciate it as fully as we might. Everybody in this audience obviously knows that dietary restriction increases lifespan, slows down age-related diseases, and everybody, I presumably knows that that high caloric availability in the form of either diabetes or obesity they all shorten lifespan. But I consider that a bit mysterious, even though we all know it. Why should too little nutritional resources be toxic, be protective, and too much nutritional resources be toxic? So that's kind of what I'm out to try to address, and that's why I feel that obesity and diabetes and dietary restriction are potentially all related. And that was just a hypothesis at one point, but I hope to convince you that there is good reason to think these are mechanistically related. So that's one question we'd like to address. Another question, of course, is why does obesity increase with age? And also, why has obesity been increasing over the last 20 years? This is an old slide, but you can still see that very dramatic increase in obesity at all the lifespans. I mean, at all the ages, and of course there's a dramatic increase with age as well, and this has just continued to increase. It may be leveling off in the last couple of years, but still, basically, the increase in obesity leading to the increase in diabetes is very likely the greatest health threat of the 21st century. So it's obvious, this was not the case when I first started this line of research, but it's turned out to be. So it's good for my research program, even if it's bad for the world. So hopefully we'll do something about that. Another interesting paradox, potentially, seemingly a paradox anyway, is that dietary restriction produces many of the same effects as genetic obesity, at least in the hypothalamus. So that seems a little strange, right? You would think that they would sort of be opposite, but they're not the opposite. As I'll show you in a moment, they're basically largely the same, and in particular, reduced hypothalamic POMC is characteristic of both dietary restriction and genetic obesity. Not only is it, so that's a kind of a paradox, but the question is there a reason for that? And I think it's not a coincidence. So we will try to address that mechanistic link. So I just want to set up an overall conceptual framework here, which I will then come back to and explain the data to support it. But the basic idea, hypothesis that I started out with is that to some extent glucose metabolism is contributing to age-related impairments, just as it clearly contributes to diabetes and diabetic complications. And furthermore, it's not just that glucose metabolism is driving these impairments. A very sort of unresolved aspect of diabetic complications, at least, is that they are relatively slow to develop and relatively difficult, or not, I'll show you, not impossible, but very difficult to reverse. So in diabetes, if you completely correct blood glucose, that does not correct the damage that has occurred. And furthermore, that damage takes a while to accumulate. So what is the nature of that clock mechanism? And of course, in principle, we have the same problem with aging. What exactly is the nature of the clock mechanism that drives slow, progressive impairments that are apparently irreversible? And Jim and I encountered a kind of a similar question in our graduate research. We were doing a similar project. In fact, I was very inspired by Jim's work and basically followed in his footsteps. But basically, we had evidence that there was a similar clock mechanism that was driving mouse menopause. And that's an oversimplification, but basically one of the things that I came away with, at least, is the concept that there is a cumulative effect of estrogen on estrogen-induced genes that gradually increase. If they're estrogen-induced genes, they become increased. Estrogen-inhibited genes become persistently inhibited, and that persistent effect on gene expression is what was actually driving the age-related impairment. I won't go into those studies, it's ancient history now, but it motivated me to look for the same kind of phenomenon with glucose, and as I'll show you, we have now demonstrated that that's the case. But specifically, the hypothesis is that cumulative and persistent effects of glucose on hypothalamic glucose-sensing neurons is contributing to the process of aging, and the dietary restriction works in part by reducing those effects of glucose. So I hope to show you some evidence to support that hypothesis, which at the time I hypothesized that was really pretty much one of those annoying theories of aging that so many are. It wasn't really based on very much evidence. So that's the hypothetical framework. Let's talk a little bit more specifically about reality here. So the part of the brain that I'm talking about, that I'm suggesting mediates many of the protective effects of dietary restriction, is the base of the brain in the hypothalamus, and specifically in the part of the hypothalamus called the ventromedial hypothalamus, also formerly sometimes called medial-basal hypothalamus. And certainly this is the part of the brain that mediates basically many of the effects of estrogen on reproductive function. But this part of the brain also regulates many neuroendocrine systems, including growth hormone, glucocorticoid secretion, and so on. And most importantly, this is one reason that I got very interested in this part of the brain as a potential mediator of aging, and certainly that was greatly inspired by Tuck Finch, who is a mentor to both Jim and I. And so basically, this part of the brain got most famous very early on when basically nobody knew what any part of the brain did. But this part of the brain was implicated as early as 1850 in regulating energy balance. That's because it was discovered that tumors could develop. Basically, there's a certain syndrome where people would get obese, their endocrine systems, especially their sexual systems, would shut down, and then they would die, and it was discovered that they had tumors in the hypothalamus. This was actually first clearly reported in 1900, but it had already been suspected before then. So that led to the hypothesis that this part of the brain regulated body weight, and that turned out to be subsequently demonstrated very clearly in the 1940s when stereotaxic surgery was introduced, that lesioning this part of the brain would robustly cause obesity in basically every animal that was ever looked at, and of course, including humans. This lesion is actually not an electrolytic lesion. It's a lesion produced by an IP injection of gold thioglucose. And it just happens that this particular chemical, for reasons that we won't discuss today, is highly trophic for this part of the brain. makes a nice hole, trophic and toxic, and it makes a very nice specific lesion here, and these animals get obese. So furthermore, if you eliminate the glucose moiety or remove insulin, this does not occur. So this led to the hypothesis that there are glucose-sensing neurons in this part of the brain that are necessary for basically energy balance and glucose regulation. And that's now been demonstrated quite clearly to be the case, although it's not quite as simple as it was thought to be 100 years ago.
So,
yeah.
So the model is you administer the gold vial glucose, and then you do something to manipulate glucose percent.
You could do that, but you don't have to. If you just inject IP gold, this is just a simple IP GTG, that's it. The animals get obese, hyperglycemic, the whole thing. If you manipulate something like you manipulate the glucose moiety or you eliminate insulin, this does not work anymore. So that supports the hypothesis that there's an insulin-dependent glucose sensing mechanism. I guess maybe you're curious, do we have any idea? My hypothesis is there's a specific glucose transporter in these neurons that make them sensitive to glucose and that makes those neurons sensitive to GTG. We could talk about the evidence for that hypothesis. It's only a hypothesis for now. But I think it has to do with their glucose sensing properties. All right, so for a long time, we really didn't have very much traction on how these neurons regulate energy balance. But one of the first breakthroughs, I think, was the observation that fasting and genetic obesity both reduce the expression of this gene, pro-opioid melanocorticotropin, which at that time was known to be in the hypothalamus, but nobody knew what it did. So we showed, and another lab showed at the same time, that fasting and genetic obesity both reduce the expression of POMC in this part of the brain. Actually, POMC is basically only expressed in a few neurons in the ventromedial hypothalamus and also a few in the brainstem. But the ones in the brainstem probably don't play such an important role. So this is just some of that evidence. And so basically, fasting, as you see, produces very marked decrease in expression of POMC. GTG also greatly reduces POMC. And then these are genetically obese DB mice, which even in the fed state have very low levels, actually further respond to fasting, and GTG is about the same. So Jim had actually already shown, interestingly enough, that hypothalamic POMC decreases with age before we did that study. So we replicated that. You did that in mice, right? Yeah. So we replicated that, and we saw the same thing. So very impressive decrease with age, which we think might contribute to the development of obesity with aging, although we really haven't demonstrated that. However, what we have demonstrated is that, but I mean, lots of things are regulated by fasting. Lots of things are changed in genetically obese animals. Obviously, the question is this functionally significant? And so we addressed that by genetically engineering OB mice to basically put the POMC back into the brain at normal levels. And we saw that it partially corrected the obesity, about 50% correction of the obesity, as you can see from the picture here, and the body weights. And partially corrected VO2 max, so metabolic rate, which is reduced in OB mice. But the most striking thing was the POMC transgene completely corrected blood glucose, so hyperglycemia. So these are the OB mice, these are the transgenic mice. And we pair-fed the mice, and that had no effect. So what that means, OB mice, of course, do eat more than wild type, but even when we pair-fed, that had no effect on blood glucose. So it seemed, very surprisingly, that these hypothalamic neurons are directly regulating peripheral glucose homeostasis. This is a glucose tolerance test, so this is a more demanding, that was just basal levels of glucose, more demanding analysis, but you can see the glucose regulation is completely normalized by the POMC transgene. And the reason, so an insulin resistance, which is very high in OB mice, was partly, although not quite completely corrected, but still very impressive correction. Now, how do hypothalamic neurons regulate peripheral glucose homeostasis? It was completely mind-boggling at the time. But we found that the transgene actually completely corrected a lot of genes in the liver that would promote glucose production. Yeah?
What was the promoter for?
There was a neuron-specific enolase. So it was not expressed in the precise way that POMC is normally expressed. However, it presumably only is acting on the normal receptors. And we found that the transgene by itself had no effect in wild-type animals. It also, by the way, corrected a lot of changes that occurs during fasting as well. So basically, somehow these POMC genes appear to be, POMC neurons are regulating hepatic glucose production. So this just promotes hepatic glucose production. This actually promotes hepatic insulin resistance, and that's also completely corrected. So we didn't know how, we still don't really know how hypothalamic POMC neurons regulate peripheral glucose homeostasis. We assume it's through sympathetic nervous system. That's still, we're still working on that. But anyway, it clearly does. That's the point I want to make. So the reason I want to make that right now is that a lot of our hypothesis depends on the mechanism by which hypothalamic neurons regulate peripheral glucose homeostasis. Okay, so but there had been a few studies that, yes?
Do you know what the effect of your knock-in, knock-out system is on renal?
No, I have no idea. We never studied renal. It's a great idea. But I will talk about renal function later in a different model. There's a lot of things we could have done with that model, but we basically moved on, mainly because we couldn't figure out how it worked. Anyway, but there were a lot of other studies that had suggested that basically neurons in the hypothalamus regulate peripheral glucose homeostasis. These are in the ventromedial hypothalamus, and basically it suggests that VMH neurons increase glucose uptake in muscle independent of insulin. So that's going to be a very important sort of hypothesis that's in the back of our minds, is those neurons, when they're stimulated by glucose, stimulate glucose metabolism in the periphery. And this is very well established phenomenon as well. If you block glucose metabolism in these neurons, that produces a massive peripheral counter-regulatory response. Blood glucose goes through the roof, epinephrine, glucagon is released, and glucose metabolism is reduced. So, and with that, you can clearly demonstrate this is specific to glucose metabolism in these glucose-sensing neurons. So clearly, these neurons can sense glucose. We have a whole program showing how these neurons sense glucose. I won't talk about that today. But they clearly sense glucose in the same way the beta cells sense glucose. And that has the effect of stimulating glucose utilization in the periphery.
Do those animals get fat?
Which animals? This is all acute. But you would expect that. That's a great question. We have not been able to do that experiment because they quickly desensitize to the 2-deoxyglucose. However, I won't talk about this, but we have shown in C. elegans that 2-deoxyglucose does induce obesity. And that's through a serotonin-dependent mechanism. I'll talk about a related mechanism when we get to it. And this just is another kind of the same thing, but not just glucose, but leptin as well in the ventromedial hypothalamus, stimulates peripheral glucose metabolism. And we have shown, which I'm not going to talk about, but basically leptin and glucose both activate the same neurons, and they're both required for the activation of those neurons. And leptin reports nutritional resources from fat, so that's kind of long-term nutritional resources. Glucose reports very acute nutritional resources. Obviously, both glucose and leptin are reduced by dietary restriction. All right, so it's a bit, definitely, I didn't even really believe this when I first proposed it. It was just kind of a model for me. But it's asking a bit much to suppose that just a few neurons in the hypothalamus mediate all the protective effects of dietary restriction. And I wouldn't even say all, but even a substantial amount. But that, I started becoming a little bit more willing to accept that hypothesis when Guarente reported that just 2 neurons in C. elegans mediate the protective effects of dietary restriction on lifespan in C. elegans. So, and these neurons are very interesting neurons, and they very possibly correspond to something similar to hypothalamic neurons. Glucose-sensing hypothalamic neurons. We don't know that. But those neurons also regulate peripheral, basically systemic innate basically immunity through an unfolding response. I'm not going to talk about that anymore, but probably a lot of you have thought a lot about and heard a lot about the role of inflammation in aging. And so interestingly enough, the same neurons that apparently control responses to dietary restriction also control inflammatory-like processes. And by the same token, similar neurons in Drosophila, those are the insulin-like peptides that produced by Drosophila, also appear to mediate many of the protective effects of dietary restriction. And we had previously shown that the glucose-sensing neurons in the hypothalamus have many elements in common with the pancreatic beta cell, including especially the pancreatic form of glucokinase. Now, they don't express insulin in the adult, but they actually do express insulin during development. So we think that those hypothalamic neurons are very similar to these DILT neurons in Drosophila. And we could talk about actually the relationship between glucose-sensing neurons in the hypothalamus and beta cells. There's a lot of similarities, and I think they probably came from the same place. But in Drosophila, the insulin-like peptides are actually produced by the neurons. So that raises the question, are these hypothalamic neurons basically the equivalent of the ASI neurons? And do they mediate protective effects of dietary restriction during aging? Well, that's a question. I'm not going to answer that question today, but I will show you a lot of evidence that's consistent with that hypothesis. So how did we go about trying to answer this question? Well, we used a genetic approach not all that different from what Jim used, and probably not by coincidence, because Jim and I are both mousers from way back, and we love mouse genetics, and so we would like to leverage mouse genetics. And so we did a study in which we looked at the expression of genes specifically in this ventromedial hypothalamus in different strains of mice that had different lifespans. And we just basically screened using gene expression for genes that would predict lifespan. Now, basically, this should not have worked. I had reasons to think it might, and that it wouldn't be that hard to do, but that turned out not to be entirely correct. But it was hard to do, but it did work. So then we went ahead. So we can't screen every gene. We just screened a bunch of genes that we thought that we had evidence were regulated by nutritional state in the ventromedial hypothalamus. And then we asked if they would predict lifespan, and then we asked the question, in C. elegans, if we blocked those genes, would that prevent the protective effects of dietary restriction? So basically what came out of that screen, and we screened over 100 genes, mainly transcription factors, that looked like they were regulated by dietary restriction. And the winner was Creb binding protein, CBP, which we published in 2009. And basically expression of CBP in the ventromedial hypothalamus, kind of only, because we looked at a lot of other tissues, and it did not correlate with lifespan, but VMH, CBP correlated with lifespan. This is lifespan under normal circumstances, and then published in different sources. So that's very surprising, maybe, because no single polymorphism could possibly account for that much variance in lifespan. So this is obviously, the level of CBP is determined by a lot of polymorphisms. So these are different strains of mice. So C57s live the longest, AJs are the shortest, and some strains in between. So CBP also showed some very nice regulatory behavior. So basically, it's inhibited by diabetes and also inhibited during aging. And so those were the kinds of properties you'd expect of the kind of gene we were looking for. Because basically what we're looking for is genes that regulate glucose metabolism and are regulated by glucose metabolism. Now, actually, up until that time, CBP was not really implicated in that way, but we did find that it was regulated by glucose in the direction we'd expect. So, yeah.
Was that correlation based on measurements of CBP in young?
Young, only in young. Yeah.
And then it disappeared.
We don't have that many old animals. So all the lifespan data are basically based on reported lifespan of these strains. That's one reason we don't have that many strains. But actually, as you'll see later, those were reported in the literature. Jackson Labs has gone back and done the same kind of studies, and we get the same correlation. Now we do it with median lifespan, but same deal. So anyway, another gene that we looked at was HSF1, which regulates, as you know, stress responses, and that also showed the same kind of decreased with age and diabetes, but it did not correlate with lifespan. SATB1 is another gene that is a CBP binding protein that showed similar behavior, also did correlate with lifespan. But FOXO3A, for example, which is implicated in the insulin pathway, does not show this kind of regulation and did not correlate with lifespan. So correlation, nobody cares. Believe me, nobody cares about correlation. So we had to go to C. elegans to do the functional assessment. So the great thing about C. elegans is pretty easy to manipulate gene expression. So we showed that we could, so first of all, we showed that CBP is in fact induced in whole body C. elegans by dietary restriction. We used several different modes of dietary restriction, got the same result. We could inhibit CBP by about 50% with RNAi. This is just bacterial feeding. So I'd just like to point out two things. It's not complete inhibition, first of all, and second of all, it's only in the adult. So that's the nice thing about RNAi. You can just give it in the adult. So they develop fine, and then we give them CBP RNAi, and it worked pretty nice. So we inhibited it, and more importantly, we inhibited its induction by dietary restriction. And so this is kind of the bottom line there. Dietary, this is a survival curve. Dietary, this is the controls. They die pretty much maximum age of 21 days. And then dietary restriction, in this case bacterial dilution, roughly doubles the lifespan. And inhibition with CBP completely blocks the protective effects of dietary restriction on lifespan. Now CBP is a transcriptional coactivator, and it also has a histone acetylation transferase function, so a HAT activity. So that suggested that perhaps part of the protective effects of CBP and therefore dietary restriction are mediated through enhanced acetylation. So to test that hypothesis, we use an HDAC inhibitor. We were talking about this at lunch. And luckily, the HDAC inhibitor we used was sodium butyrate, and I'll come back to why that was a really good choice later. But as you see, in fact, this is a well-known inhibitor of HDAC. Let me explain that a little bit. HDACs they do the opposite of HATs. So they take acetyl groups off of the proteins that HATs acetylate. And so the hypothesis was if we inhibit those activities, that will enhance the effective activity of CBP. And we showed that that was in fact the case. So sodium butyrate did in fact increase lifespan. And if we inhibited CBP, that was completely prevented. So that suggests that the relevant HDAC activity was specifically on the HAT activity of CBP. And we showed the same thing. We have nice models of different human diseases and C. elegans. So we have a model of Alzheimer's disease. So this is the actual human gene that causes Alzheimer's disease in people. We put that into C. elegans, they get paralyzed at about the same time. So this is that famous relationship. So they get paralyzed about 2/3 of the way through the lifespan, just about the same time you get Alzheimer's disease. And so we showed that that was protected by dietary restriction, slowed down by dietary restriction, completely dependent on CBP. We also did the same thing with sodium butyrate. So they get paralyzed with age. Sodium butyrate slows that down. And if we inhibit CBP, that protective effect is completely prevented. In fact, the paralysis greatly accelerates. So this suggests that in this particular model of proteotoxicity, I'm not going to say it's a great model for Alzheimer's disease. We could talk about that. But certainly it's a model for age-dependent proteotoxicity, and it's it's basically improved with HDAC inhibition dependent on CBP. Okay, so CBP seems to be important. How is it working? So as I suggested early on, the hypothesis was that glucose metabolism is driving aspects of the aging process just as in diabetic complications, and that dietary restriction works in part by reducing glucose metabolism. We had a lot of preliminary evidence that, in fact, what dietary restriction does is switch glucose metabolism away from glucose metabolism and toward beta oxidation, but certainly many other alternative fuels. And Dr. Masro and I have had a long-standing discussion about whether this is true over 24 hours and everything else, but certainly in certain tissues, it's appeared to be the case regarding at least gene expression. And so we have now corroborated that, at least with fasting, that in the hypothalamus, the gene expression clearly indicates that after a 48-hour fast, the genes clearly indicate that glucose metabolism is reduced, alternative substrate metabolism is increased, including lipid oxidation, which until recently the brain was not thought to support lipid oxidation, but the gene expression profile clearly indicates that it is. That's published, and you can see the evidence for yourself. So what is the big deal about glucose metabolism and this dietary restriction switch? I'm not going to get into this because it's basically hypothetical, but I'm just going to say there's lots of reasons to believe glycolysis is particularly toxic relative to other forms of making ATP, and dietary restriction actually produces a lot of changes at the gene expression level that could plausibly be protective. And I'm just going to point out a couple of them because it's a little complex, and even though it's all plausible, it's still hypothetical, so I don't want to dwell on it too much. But I'll just point out a couple of things. So basically, dietary restriction actually enhances lipid oxidation, which should turn over lipids that have that maybe have developed oxidative damage. Actually, this should be amino acid oxidation should be up with dietary restriction, and that will actually prevent the accumulation of oxidative damaged proteins. And dietary restriction increases the ratio of NAD plus to NADH, and that theoretically would enhance sirtuin activity, which may be protective, and would also presumably inhibit CTBP activity, which is another transcription factor, which is the anti-CBP transcription factor. And I'll show you in a little while evidence that this really, well, definitely regulates lifespan or impacts on lifespan. So we showed that same pattern. That was all from mouse hypothalamus, but we showed the same pattern in C. elegans with dietary restrictions, his whole body. So in C. elegans also, dietary restriction reduces, for example, phosphofructokinase, which is a rate-limiting step for glycolysis. But if you inhibit CBP, that actually increases the expression of this glycolytic enzyme. Conversely, dietary restriction induces CPT1, which is the rate-limiting enzyme for beta oxidation, lipid oxidation, and CBP inhibition completely wipes that out, really reduces the expression of that gene. So this is consistent with the hypothesis that dietary restriction induces CBP activity. That causes the switch from glycolytic activity to alternative substrates, and that inhibition of CBP prevents that switch, consistent with the hypothesis that that switch does contribute to protective effects of dietary restriction, but does not prove that hypothesis. Yeah?
So if you slow down the glycolytic pathway, so what happens to the mitochondrial function? So there's no go to the acetyl-CoA to kissing cycles?
From glucose carbons.
From glucose carbons.
Yes, but then we have alternative substrates like beta oxidation and especially ketones. Right, yeah. So I would like to emphasize this point. I'm not talking about reducing metabolic rate. If anything, we may be increasing metabolic rate as far as oxygen consumption, because to get the same number of ATPs from lipids as you get from glucose, you have to use more oxygen. So you probably are aware of evidence supporting that actually you increase respiration in certain forms of dietary restriction to increase lifespan. So this is if anything, it's ATP neutral or maybe even increases respiration. Yeah? Right. That was that first graph I showed. Sorry. Yeah, and it also blocks all the protective effects on proteotoxicity. Yeah?
Charles, sorry. Can you go back a slide? So this was done in worms, or
this was done. Well, this is from a lot of different data, lots and lots of data. I have a paper I can send you. So this is really more or less from the literature, most of this stuff. What this is done in mouse hypothalamus. But for example, this business, if you inhibit complex 1, 3, or 4, or 5, that extends lifespan. That was done in worms.
But that if you're using fat, right, your acetyl-CoA, it's going to be put a lot of NADH.
In the mitochondria, but not in the cytoplasm. And you're right, but nevertheless, you get NADH, but you get more FADH2. And FADH2 is oxidized at complex 1. Sorry, 2. So that's the idea, is by shifting toward the utilization of FADH2, even though it's less efficient, it uses complex 2.
That's not correct.
Oh, really?
Yeah.
Okay. Well, so as far as I know, it's correct. Guarente thinks it's correct, but I'm happy to be corrected on that point.
Because the succinate is coming in. That FADH2. I'm not even sure that that's coming in via complex 2, right? And I think
FADH2?
That's a bound moiety.
Well, let's talk about this offline. I said I don't want to dwell on this too much because it's really based not on our work, but the literature. And certainly with regard to complex utilization, there's a lot of wiggle room in those data. So this is just to give you sort of a general notion of why that may be true. But as I say, Guarente pretty much made the same point at about the same time. So I guess we could both be wrong. It's very possible. God knows. So anyway, so that's the basic hypothesis. But again, this is not proven. So the question is, how would dietary restriction be influencing CBP activity? Because there's no obvious link. And also, for reasons I won't go into in detail, as I suggested, NADH seems to be an important component of the toxic effects of glucose and some signaling effects of glucose. So there, of course, is the sirtuin story, which is consistent with the role for NADH, NAD+. But there was another transcription factor that I got very interested in, which is called CTBP. And the reason I got interested in it is it's actually regulated by NADH, which, as I said, is really, NADH in the cytoplasm and the nucleus really only come from glycolysis. So that suggested to me and we had a lot of other reasons to suspect NADH also, both for signaling and for toxicity. So this paper came out demonstrating specifically in the brain that the NADH actually binding domain of CTBP mediates the effects of glucose metabolism, in this case 2-deoxyglucose, on brain gene expression. So this really suggested to us that CTBP might be a plausible mediator of the effects of nutritional status on the CBP complex, because CTBP interferes with effects of CBP. CTBP is a transcriptional inhibitor and co-repressor, and CBP is a co-activator. So we did the following study, and I apologize if you probably can't read most of what's on here, but I'll give you the general point of it, and then I'll get into some more details. So these strains of mice obviously don't differ just by their lifespan, but they also differ in a lot of other ways, obesity and blood glucose and many other things, and which we characterize those. We could characterize all those ourselves. So we asked the question, do these genes also predict these phenotypic responses? Remember, the idea here is that there may be an intimate relationship between obesity and blood glucose and lifespan. So we looked at those parameters, and this is what we found. We found that, in fact, the same genes predict lifespan and blood glucose and weight gain on a high-fat diet. So this is CBP, for example, and this is the lifespan data. Now, this is new data on median lifespan, but basically the same correlation. And blood glucose. Now, this is standard condition blood glucose, just basal blood glucose on a low-fat diet. The same hypothalamic gene predicted blood glucose positively. That was a big surprise to me. And also predicted obese phenotypes on a high-fat diet, total adiposity, blood leptin, blood insulin, and weight gain. They're all positively and significantly correlated with CBP expression in the ventral medial hypothalamus, but no other tissue that we looked at. Conversely, CTBP, the anti-CBP, is inversely related to blood glucose, lifespan, and markers of obesity. And similar for the other genes, but we won't really talk about that right now. So, but the point is that the CTBP and CBP both predict lifespan, positively predict blood glucose, and weight gain on a high-fat diet. So 2 points are, first of all, CTBP and CBP correlate in the opposite direction, just as you would expect to by their function, and that obesity and blood glucose are positively associated with with lifespan on a low-fat diet. I was very surprised about that at first, but I think we can make sense of it. So this just shows you the data in a little more detail. So this is blood glucose and hypothalamic CBP. So again, nobody cares about correlation. So if anything, it's almost bad if you get a correlation because that's just so statistical. So we were luckily able to do assess some of these phenotypes in mouse models. So for example, we actually have a conditional flox-CBP that we can knock out just in the adults. This is with tamoxifen inducible. So basically, so we focus just on the two extreme strains, C57s and AJs. And so this is actually very well known that C57s have elevated blood glucose compared to AJs. And if you put them on a high-fat diet, C57s, of course, are famously very sensitive to a high-fat diet. They get very glucose intolerant. AJs basically are fine on the high-fat diet, even though they actually are consuming just as much of the diet. But they do not get glucose intolerant, and for that matter, they don't get obese. And when we knock CBP out just in the adults with tamoxifen, this is C57s. They normally this is on a high-fat diet. They normally get very glucose intolerant, as you saw. Knocking out CBP in the adult only completely protects them from glucose intolerance on a high-fat diet. And this is on a low-fat diet, just looking at blood glucose. A VMN-specific ablation of CBP, this is with the SF1 promoter, reduces the blood glucose to almost the level that you see in AJs. This is, again, C57. That may not seem like a big difference to you, but blood glucose levels are really very reliable. And this is a really, this is a very impressive decrease in basal blood glucose. Yeah. Yeah. For the ventromedial nucleus, mainly, yes. It did not hit the POMC neurons. I didn't want to get into complications, but the VMN neurons, the SF1 neurons, regulate POMC neurons. So they kind of behave the same way. Anyway, however, we actually are focusing more on the VMN neurons as being more relevant to blood glucose regulation, but they also regulate POMC, so it amounts to the same thing. So anyway, so this is how CBP correlates positively with weight gain on the high-fat diet. Again, only hypothalamic CBP. So again, we could knock CBP out of the whole body postnatally, and that partly, although certainly did not completely protect against weight gain on a high-fat diet. So Creb1, which is obviously one of the many transcription factors that recruit CBP, also positively correlated with weight gain on a high-fat diet. CTBP2, on the other hand, negatively correlated with lifespan and negatively correlated with blood glucose. And that's just weight gain on a high-fat diet was negatively correlated. So we did the obvious study, which is to inhibit CTBP in C. elegans. The obvious prediction there is that should increase lifespan, sort of mimic the effects of dietary restriction. We got that, but as we were preparing to publish it, it was published for us in PNAS, but that's fine, it's good. So they showed, in fact, that inhibiting CTBP does increase lifespan, and very importantly, that required the expression of a triglycerol lipase. So that suggests that the protective effects do, in fact, of CTBP inhibition do, in fact, involve somehow beta oxidation. So we got scooped on the lifespan, but we didn't get scooped, so far at least, on the obesity side of CTBP. So we showed, I mentioned earlier, we can induce obesity in C. elegans with 2-deoxyglucose. Can't do it in mammals because they desensitize. But we saw the same thing if we inhibited CTBP. This is just with TLC. You can see that inhibition increases it. This is the quantification. And we could also do that with fluorescence. But interestingly enough, we could reverse that obesity with serotonin. So that demonstrates that it's a neuroendocrine phenomenon. Okay, so again, focusing just on AJs versus C57s, looking specifically at the difference between those two strains, of course, this is consistent with the correlation that C57s, B6 C57s show elevated hypothalamic expression of CBP and reduced expression. There's two CTBP isoforms. They behave very similarly. They're both reduced. CREB1 is induced, and SATB1 is also induced. Expressed at higher levels in CBP, C57s versus AJs. So what's the functional significance? Remember, the hypothesis here is that CBP basically inhibits glucose metabolism and promotes lipid metabolism. And the implication of that, therefore, is that actually the hypothalami of C57s would be less sensitive to glucose than the hypothalami of AJs. So over the years, we've developed over 100 markers for glucose action in the hypothalamus, in the ventromedial hypothalamus. So just using a few of those, what we can show is that, so these genes are responsive to glucose both in vitro and in vivo. And basically, we could show that those genes are in vitro regulated by glucose in AJ mice, but under those same conditions, they're insensitive to glucose in C57 mice. And that's basically true for all these genes. So now that's the genetic comparison, which is very interesting. But as we all know, actually, aging is not mainly genetic. It's mainly, well, not genetic, within a strain, within a species, although, of course, across species, aging is highly genetic. Well, C57s are the same way, of course. C57s don't all die on the same day. There's a huge spread of when C57s die. Interestingly enough, that's also true with diet-induced obesity. These are basically all genetically identical mice, but they have a huge range of response to diet-induced obesity. This is just an example, but it's very typical. Some mice get almost double their body weight on a high-fat diet, and some mice basically don't gain any weight on a high-fat diet. So the question is, do the same hypothalamic genes predict this variation, even though they're in genetically identical mice, as predicts the variation in, across genotypes? So first of all, just to give you a little more information here, these are all C57s. This is basically the intake the grams intake per day, and this is how much weight they gain. And what you can see is in C57s, basically the main difference in how much weight they gain is how much food they eat. That's actually not true between strains, but within a strain, it's mainly how much they eat. But the point is, well, you might think that's obvious, but why should that be? Why should one particular genetically identical mouse consistently eat more than another one? And this just shows that that's the case. So basically, if they eat a lot on the second week, they eat a lot on the first week. It's a stable personality trait that mainly is what accounts for their different weight gains. And that's also predicted by expression of CREB1 and CBP. So this is the obesity-resistant mice. So basically, we took 100 mice, put them all on a high-fat diet, then found which got obese, which did not get obese, took the top 10%, the bottom 10%, looked at their hypothalamic gene expression, and the mice that got obese were the ones that expressed the highest levels of CBP and CREB1. Yeah.
Have you confirmed any of these things with protein levels?
No. So I will get back to that. I'll talk about protein levels later. I'm happy to talk about it, but not now. Yeah. We didn't do the whole group. I mean, 100 animals, please, give me a break. I suppose we should, but we just took the extremes. But I just want to emphasize, these are genetically identical mice. So basically, so these variations are due to some kind of epigenetic differences. I'm guessing developmental differences, but whatever the differences are, the point is, even genetically identical mice don't all have the same gene profile. Yeah.
Okay, so you've got tomogenic animals, but what about the immediate birth effect of feeding?
Absolutely.
I mean, and that may be behaviorally controlled by the mother.
Absolutely. I'm not going to weigh in on the reasons that are different, other than the fact that the same genes are implicated in both cases. Could be birth order, could be how much they ate, how big the litter was, all kinds of. Actually, other people have studied this phenomenon, and they have not been able to pin it down to any particular litter size or anything else like that. But we haven't done those studies. We just did the gene expression. But it could be any of those things. Could be development, could be birth order, could be anything. Yeah. No, okay. So the question I would love to answer is, would this also be the case with lifespan? But it's kind of hard to do that with lifespan because, you have to wait till they're dead. So, tricky. Anyway, so how is CBP then working here? And so over the years, my attention has I've started focusing my attention more on the PPAR family. And the reason for that basically is that the one transcription factor in all of, I guess, nature, but certainly in animals, whose job it is to switch metabolism away from glucose metabolism and toward beta oxidation is the PPAR family, particularly PPAR alpha, but also PPAR gamma. And the PPAR family recruits CBP, but that's inhibited by CTBP. And the PPAR family binds to DNA elements, PPAR response elements, and the kind of classic genes that they induce are CPT1A and PTK4 in the liver. It's classic. And what that does is CPT1A promotes beta oxidation, and PTK4 blocks glucose utilization by inhibiting pyruvate dehydrogenase. This is very well established in the liver and the muscle media, and this is induced, by the way, by fasting, but also dietary restriction. So what happens is free fatty acids are released, they activate the PPAR transcription factors, especially alpha, and that causes this switch. So I should have figured this out a lot sooner than I did, but the reason I didn't is that I was focused more on glucose metabolism, and PPARs are mostly famous for lipid metabolism, and I hadn't really focused so much on lipid metabolism because this is the brain, for God's sake. But anyway, eventually I got the message, so we decided to look at the role of PPAR alpha, and right, well, so I'll get to that in a minute. But first, just to go back to the mouse model now. So basically, dietary restrictions, 3 weeks of dietary restriction, looking at the hypothalamus only, of course, as I said, induces CBP. This is not true for most tissues. CBP is also induced in liver, but not in any other tissue we've been able to find. But it is in the hypothalamus, and that's correlated with the induction of CPT1A, which supports the possibility of PPAR alpha, and that is also correlated with the induction of PTK4. So originally, I noticed PTK4 and CPT1A were induced by dietary restriction and fasting. That was kind of my first clue about this whole thing. So now we see that they're correlated with the induction of CBP, and also, interestingly, FOXO3A, which, as you know, is the kind of the good guy mediating the insulin pathway. And I will come, that was not known, it's still not really known that there's a relationship between CBP and FOXO3A, but you can imagine how that might be protective, and we'll get back to what's mediating that, but specifically we'll look at the hypothesis this is mediated by the ketone 3-hydroxybutyrate. So basically, CBP is correlated inversely with blood glucose, consistent with our observation that high glucose inhibits CBP expression. So now we can go to the genetic manipulations. So we could actually enhance PPAR expression in the hypothalamus with adenovirus. So the PPAR alpha knockout actually has improved glucose tolerance. This is the wild type, and if we overexpress PPAR just in the ventromedial hypothalamus, that improves. That impairs glucose tolerance, which is what you'd expect, right? Oops, sorry. And we showed that expression of PPAR alpha experimental increase in PPAR alpha about 3-fold, which is about the level of activity induction by dietary restriction, induces the expected genes in hypothalamus, CPT1A and PTK, and also not only impairs glucose tolerance, but actually improves responses to hypoglycemia. So in other words, again, suggesting that that is reducing sensitivity to glucose. And furthermore, expression of PPAR alpha after gene transfer correlates with blood glucose, correlates with CPT1A. CPT1A correlates, but not quite significantly, with blood glucose. So we've now extended some of these observations to other models of proteotoxicity. Now this is in mouse. So we have a mouse model of Huntington's disease. So these mice have very poor behavior on the rotarod test. And if we give them, this is dietary restriction, actually reverses their impairments. This is every other day dietary restriction. Fasted versus fed, fasted versus fed. What I like about this is it's not caloric restriction. because they compensate every day. But they do have lower average blood glucose. Clearly, they have a lot more exposure to ketones, which I'll get back to in a minute. And that completely normalizes their functionality, even after they had impairments, and induces CBP. Actually, CBP is more induced in the transgenic mice for some reason. So what's the actual role of PPAR? Well, C. elegans only has one PPAR, but we can knock it out. And so that's the nice thing about PPAR alpha, at least. You can knock it out and the animals are viable. Same is true with worms. And so this is the effects of dietary restriction. And the PPAR alpha knockout almost completely, not quite, but almost completely prevents the protective effects of dietary restriction. And that was also true. We have a worm model of Huntington's disease. So this is, again, the human Huntington's gene causes paralysis. Dietary restriction is protective in that model, and knocking out PPAR completely prevents those protective effects of dietary restriction on this model of proteotoxicity. So what about CPT1A itself? And so basically, yeah.
Back to the mouse.
Yeah.
Did you do every other day restriction because of the facility of doing it, or for a particular reason, or did it?
Yeah, actually, we did it because I'm very interested in that model. So I just wanted to know if we could employ it to try to understand mechanism. But actually, to be honest, it had already been done in a different model of Huntington's. So I had a little bit more security that it would work than I might have otherwise.
But did they completely compensate for food intake on the days they ate food?
Yeah, as far as we can tell, yeah. So anyway, so what happens if we overexpress CPT1A, again in the ventromedial hypothalamus? Basically, it produces obesity, just as we would have predicted. And partly that's due to increased food intake. So again, the idea is you're shifting toward beta oxidation and away from glucose, which glucose metabolism is really good for the short term. It keeps you thin, keeps your blood glucose down, but in the long term, we think is toxic. Yeah, I think I already showed that, sorry. Oh, I'm going backwards, that's why. So the question is, what's the mechanism of the overexpression? And in this particular case, the way we do this, the overexpression is basically confined to the ventromedial nucleus, the SF1 neurons. POMC is outside of that field. But we know that those neurons regulate POMC, at least activity. And interestingly enough, when we overexpress CPT1A, that actually did not inhibit any other genes in the ventromedial nucleus or do anything to any of the other genes except for POMC. POMC is inhibited. Now that's outside those neurons. So, but we haven't actually proven that, but we are pretty confident this is an indirect effect of these VMN neurons regulating POMC. SF1, so one thing you're always worried about these kinds of studies, if you damage the VMN, the animals get obese. So you're always worried about that. So we have a lot of ways to assess that, but SF1 is a really good one, because if you damage the VMN, you decrease SF1 expression, but there's no decrease, if anything, an increase. It's a really interesting phenomenon here. Surprisingly enough, if you overexpress CPT1A, that actually induces PPAR alpha and PPAR alpha target genes. It itself is a PPAR alpha target gene. So what that means is that there's a hysteretic effect here. It's a positive feedback, and this has actually been established in other similar models just with response to fasting. So it means that the system resists change, and it kind of perpetuates a steady state. So we could talk about the significance of that, but it was unexpected, but very cool, because it actually can explain a lot of other phenomena. So I use the term hysteresis. So basically that means a kind of like a gene memory. And I mentioned early on that this was something we were looking for, and what we found, so this was actually very theoretical, to tell you the truth. But until this paper came out. And what this paper showed is that the lac operon shows hysteresis. So let me show what that means. So it means if you induce the lac, these are individual cells, and if you induce the lac operon by increasing levels of inducer, these are E. coli, you get more and more induction, obviously. But what's interesting is, and first of all, notice it's not. It looks linear if you take the whole population, but what it actually is you're inducing from an off state to an on state, and you're just more and more likely to be in an on state as you get more inducer. But more importantly, if you go the other way around and start at high inducer and go down, you have to go to really low levels, actually almost very, very low levels, to get complete reversal. So what that means is that there's a kind of a form of gene memory that's built in even to the lac operon, and that's exactly what we were proposing for estrogen and also glucose. And glucocorticoids also may have the same kind of function. So is this true with glucose? And we showed that it is. So we've shown this in other cell types, but we specifically used Schwann cells here because they're implicated in diabetic complications, neuropathy, and we can cultivate them for long periods of time. And I had my basic guess, well, it's very interesting. My guess was that it would take kind of a long time to develop this hysteretic effect because it takes a long time to develop diabetic complications. My hypothesis is what happens is every time the cell sees glucose, it pops up some of its genes probabilistically, and it takes a lot of individual exposures to get all those genes, all those cells in that on state. So we basically cultivated Schwann cells for 8 weeks at high glucose 25 millimolar versus low glucose 5 millimolar. And what we found is basically essentially every gene in glycolysis was induced by 8 weeks at high glucose, and that was irreversible. If we put the cells back at low glucose, the induction persisted. We've never been able to reverse it for as many weeks or months as we. So this shows a form of gene memory that's proglycolytic. Glucose induces a permanent increase in glycolytic machinery. We actually measured glycolysis, and it actually is induced. Interestingly, 3 genes that block glucose metabolism, because they're part of the regulatory cycle here, they're shown here, they're all persistently and greatly, especially these 2, inhibited. So this produces a massively proglycolytic molecular profile that's persistent. And so this just shows the other genes. Basically, we see that whole switch. All the glucose genes are induced. All the alternative genes are permanently inhibited. But it only works. You don't see these effects acutely, just like you don't see diabetic complications acutely, just like you don't see aging acutely. The first genes you can actually see regulated are at 4 weeks. And then by 8 weeks, you see the whole profile.
How do these glucose levels compare to?
Yeah, 5 millimolar it's a good question. 5 millimolar is fasting. 25 is way superphysiological. We just wanted to make sure we saw an effect. We've now scaled this back to 15 millimolar, which is basically diabetic levels, and we see the same thing.
A question.
Yeah.
You said permanently, so you've never seen if there's a way to switch that back?
We've tried lots of ways, but have never been able to do it yet. But I'll show you some evidence that we might be able to do it actually in vivo. But we've tried lots of things, and certainly putting them back at 5 millimolar does not do it for weeks. Yeah. Yeah, we're working on that. That's good. Hold that question. We'll get back to that. But yes, we believe that this acetylation is reduced. Actually, we looked at acetylation levels. It's not that simple. It's not like it's really annoying. The acetylation levels do not really match the expression levels and the methylation levels. We did DNA methylation. There are permanent changes in acetylation, permanent changes in DNA methylation, but they're not obviously related to gene expression. So it's not a nice little package. Yeah?
When you treat the cell for 8 weeks, is it passage? Or
yeah, of course. Yeah, of course, we had the passage. So yeah, it's not confluent. So we passage them about twice a week. So good point, because what this means is these changes they persist after cell division. So that's why we thought DNA methylation would be a good thing to look at. Well, there are permanent changes in DNA methylation, but they don't correlate with, annoyingly, with gene expression. So we're still working on that. I still think acetylation is important, but it's probably some specific part of acetylation. Okay, so, but the one epigenetic change that did seem to make sense is that the binding of PPAR, in this case gamma, to the target genes was permanently inhibited. That's what we were hoping to see, because we don't know the mechanism of that, but that is consistent with the basic hypothesis. We did PPAR gamma because basically we could. So we got to go back and look at PPAR alpha. But so we don't know what the mechanism is that's reducing the binding of PPAR alpha to the target genes. So again, to remind you of hysteresis, the beautiful thing about this hypothesis, well, this observation, is that it suggests that if you could get, in this case, the levels of glucose low enough, normal levels aren't going to do it, but if you go low enough, you should be able, maybe, to reverse maybe diabetic complications, maybe age-related impairments. Can you get glucose levels low enough? No. Dietary restriction doesn't go low enough. If you use hypoglycemia, we tried that, that kills the animal. So what to do? Well, it turns out we actually had a weapon at our disposal, but it took me a while to figure that out. So we were doing basic mechanisms of glucose sensing in the hypothalamus. And as I suggested earlier, my hypothesis was that the key signal for glucose, presence of glucose, was not actually ATP. For various reasons, we had a lot of evidence for that, but nobody believes us. So I said, well, let's change their ATP levels some other way and show that that does not mimic the effects of glucose. So we decided to do that with ketones. So we knew neurons will certainly metabolize ketones. And the thing is, you only see ketones when you're fasting. So you really don't want ketones to mimic the effects of glucose. If anything, you want them to block the effects of glucose. And that's exactly what we saw. So in this case, we're using the gene AGRP as a marker of glucose action. We have tons of them, but this is what we use in this particular case. So basically what you see is if you reduce glucose from 10 to 0.5 millimolar, that induces AGRP. AGRP is also induced in the hypothalamus by fasting and dietary restriction. So fine, it's an glucose-inhibited gene. So if ATP is the signal, and if, basically if ATP is the signal, you'd expect ketones to mimic the effects of glucose and inhibit the gene. But in fact, ketones progressively induce the gene. So they're inhibiting the effects of glucose. But it is producing ATP. So I think this pretty much rules out ATP as the key signal. That was interesting to me, but then it occurred to me, well, then maybe ketones can be used to block glucose metabolism in vivo. And then I remembered, oh yeah, our old friend sodium butyrate is like almost the same thing as 3-hydroxybutyrate. I swear to God, I did not think of this until well after these experiments were done. It was just a coincidence that we used those two compounds. So, but we did show that ketones are highly protective to neurons under oxidative stress. So this is viability of mouse primary neurons, and at high glucose, they're basically, we give them a little bit of oxidative stress, so there's about 50% viability. Ketones are highly protective. And of course, with very low glucose, they're basically completely wiped out, and ketones are highly protective under those circumstances as well. So we did the experiment to use a ketogenic diet to try to reverse diabetic complications. So as I mentioned earlier, diabetic complications like aging are progressive and thought to be irreversible. It's a big question in the field. It's called metabolic mystery, metabolic memory. And the question is, why can't you reverse diabetic complications? So for example, if you correct blood glucose completely in a diabetic animal, they still continue to show oxidative stress. What's up with that? So to really push the point home, we waited. This is both type 1 and type 2 diabetic mice. We looked at nephropathy first, because nephropathy is really easy to study from a functional point of view. You just measure albumin to creatinine ratios. So we waited until the mice actually developed basically almost kidney failure, according to their albumin to creatinine ratio. Then we put them on the ketogenic diet. And the ketogenic diet induces ketones just like dietary restriction, but without caloric restriction. And what we found is after they developed the kidney damage, after 8 weeks on the diet, they were completely normalized for both type 1 and type 2 diabetic mice. And this just shows a bunch of genes that we developed that were correlated with kidney failure, mostly oxidative stress genes, and they were basically pretty much all completely normalized. Although not all completely, so that's an interesting question. Where's Holly? Oh, for Holly, because that's my point, that we would like to know which oxidative stress is corrected and which ones aren't. So that was published. We did the same thing for aging. C57s don't really get kidney damage, so we had to use AJs, our relatively short-lived strain. So we put some mice on the ketogenic diet at about 9 months of age, and then other mice, we waited until they developed kidney damage and then put them on the ketogenic diet, and then we either completely prevented or actually completely reversed the kidney damage. These are the animals on the ad-lib-fed diet. And this is just, again, the basically oxidative stress genes that were induced with age in the kidney, and that was prevented or reversed by the ketogenic diet. We have now extended this to diabetic neuropathy, and so we see the same thing. DB mice develop diabetic neuropathy. This is a behavioral test with von Frey hairs, and after they develop the neuropathy, that's completely reversed. And we're working on the gene expression for those guys. And then finally, so one of the most striking things about dietary restriction, obviously, is its reduction in cancer burden. And this is true for, I don't know how many millions of forms of cancer this has been demonstrated for. Whether it's induced or transplanted or whatever, dietary restriction is massively protective against cancer. You probably know about the Warburg effect. Basically, cancer cells live off of glucose metabolism. So if you could block glucose metabolism, a lot of people think that's going to be very protective against cancer. How do you do that? Well, we did this in basically a model of breast cancer, which is highly insulin dependent. And so the hyperinsulinemia greatly stimulates the growth of the tumor, and the ketogenic diet really greatly prevents the growth. It doesn't completely prevent the growth, but greatly prevents it. And then finally, it basically almost completely prevented the metastasis of that in that particular model. So the hypothesis here that we were developing was that 3-beta-hydroxybutyric acid is an HDAC inhibitor, just like sodium butyrate. And so dietary restriction works by inducing 3-beta-hydroxybutyrate, blocking HDAC activity, enhancing CBP activity, and then you get the appropriate metabolic changes. So as we were working on that hypothesis, this paper just came out. So we were scooped, but it's okay, it's all good. That's exactly the case. 3-beta-hydroxybutyrate is an HDAC inhibitor, just like sodium butyrate. Well, maybe not just like, it's more specific probably to HDAC1 and HDAC2. And it induces FOXO3A. Which we showed whose induction is correlated with CBP, at least in the hypothalamus. So that's the hypothesis, and with help from some very good friends, that hypothesis is actually coming along pretty nicely. So where do we go next with this? Basically, obviously, are the protective effects of dietary restriction or the ketogenic diet that we see, are they prevented by VMH-specific ablation of either CBP or PPAR alpha? We now have, obviously, the conditionals for both of those. I have a proposal, well, I had a proposal in that was triaged, but who knows? Maybe we can get that going. They triaged it because they said it was too ambitious, and that probably was true, so we'll cut it back and see what happens. And we have the tools for that. We can also similarly ablate the ability to use ketones. So there are specific genes that are involved. We have those floxes also. So that would be very interesting if we could show that the protective effects of ketogenic diet on kidney function are mediated through hypothalamic ketone utilization. But we'll see. And we're also working on ketone derivatives because ketones themselves are not really, they're metabolized too quickly to actually use them therapeutically. Although sodium butyrate is pretty good, but it's got a lot of bad side effects. So basically, the conclusions are, I kind of just went over those, so, but I'll just say very briefly, protective effects of dietary restriction are mediated by reducing VMH metabolism of glucose. and glucose metabolism drives many age-related impairments prevented by DR. Molecular effects, profile of DR is regulated by this master transcriptional complex, which is probably opposed by NADH regulated CTBP. The same complex links the positive in the hypothalamus links the positive correlation between lifespan, blood glucose, and obesity. The molecular effects lead to this particular profile switch, which we believe is basically toxic. And the ketogenic diet basically reverses at least some aspects of glucose metabolic memory and prevents many of the many different pathologies that are also protected by dietary restriction. So if that was all too much for you, I'll sum it up as follows. Glucose behaves like Don Juan. It shows up, does its thing, then it's gone. But when genes have been kissed, some effects may persist, and the melody still lingers on. Thank you. And I would like to acknowledge the wonderful people. Hopefully you saw their great names on the slides. I try to give everybody appropriate credit, but these are just a lot of the people. Minhua Zhang and Michael Poplawski did most of the CBP work. Wei Cheng did a lot of the work in the hypothalamus and also the AGRP regulation. Linda did a lot of the CPT1A work. Kelvin Yen did a lot of the early C. elegans work. Hector did a lot of the C. elegans work in obesity. Fumiko Isoda runs my lab, and I can't do anything without her. This is my daughter who snuck into the picture. Thank you very much.
Thanks a lot. Charles, questions?
Yeah.
How different diets reduce the polyQ elegans?
Pardon me?
How different diets
Yes.
reduce polyQ elegans?
Did I do what?